Thanks guys for being here. Today I'm going to be talking about metallomic imaging and how we can use elements in our imaging techniques to unlock the insights into health and disease. So this talk kind of splits into a few sections. A little bit about what metallomics is, in case no one's actually heard of it. That's generally no one's heard of metallomics, so I start there. Then I'll talk a little bit about the instruments, focusing on imaging, and then hopefully some really exciting case studies of where we've applied this to different scientific areas in healthcare and disease, and show you hopefully some cool images and some nice data. So let's start off. Okay, so metallomics, as the name suggests, it's to do with metals. So if you split the word in half, we have metals and omics. It might surprise you that actually of the periodic table, 85% of the periodic table is actually made out of a metal. And these are sort of the fundamental building blocks of life and matter themselves. So you can imagine they're quite important to a lot of things. Around sort of 50% of all known proteins and all known enzymes in the body actually have a metal that's inside it, whether it's sort of intrinsically linked within its structure, or whether it's part of it used as a catalyst for a reaction. So you can imagine that we need these metals to survive and live. Actually, in our body, we actually only are made up of around 2.5% by mass of metals. When people say we're predominantly made out of water, they're not lying. We're mostly made out of things like oxygen, carbon, hydrogen, and nitrogen. But this 2.5% is very important. And so our body have these really complicated homeostatic methods to keep the regulations of these key metals in our bodies at certain concentrations, far beyond what I can understand because I'm not a biologist, but trust me, there are lots of different ways to keep metals to the status quo in the body. And you can imagine if you get dysregulation of too much or too little of these metals, then you get problems and diseases. On the flip side of the word, omics, this I don't need to probably introduce to this audience, but this is the study of sort of biological molecules to find out their roles in living cells and human health. Probably why most people haven't heard of metallomics is because we're kind of the new kid on the block. We were only coined around sort of 20 years ago that metallomics was first described. And the other categories of omics, things like genomics, proteomics, transcriptomics, and metabolomics. The metabolomics is always the one that people get confused between us and them. These were coined much earlier on and been much more established scientifically in the community. I like to think of metallomics as a bit of a master integrator of all of these different omic modalities, because frankly, you need all the metals within all of them. So hopefully, yeah, I'm preaching the word of metallomics to you guys here today. So a little bit about metallomics itself. I've alluded to this a little bit already today, which these are so the first category of metals that we can think about are known as the endogenous metals. So these are the metals that normally live inside you and I. We regulate them at certain concentrations, and without them, we'd probably die, or we get probably some really significant diseases and disorders. So things like iron, copper, calcium, sodium, all of those transition metals that we know are important. And yeah, so a real simple example of this is in the case of iron. If you have too little iron, we know this can cause a condition known as anemia. We have a blood test for this. When we have too little iron, we can't produce enough hemoglobin to carry oxygen around the blood, and we feel very tired and lethargic. So that's too little iron. If you have too much iron, you have completely opposite side. This is where we have too much. It causes a condition called hemochromatosis. It causes lots of DNA damage and cell death and a handful of different problems. So that's one metal, too much and too little. And you can imagine there are lots of metals and loads of different conditions they can cause, of which here are some of them. So I'm not going to go through every single one of these because that would be a very boring talk. But this is just to highlight some of the key metals, sort of scientific interests that we have in metallomics at the minute, and some of the active research projects that I'm doing here at the London Metallomics facility. So just to highlight a few, and I'll go into a few in detail later, but we have key things like cancer, cardiovascular disease and heart disease, neurodegeneration and bits like that, and also infection. So lots of metals are different and important, and this is definitely not an exhaustive list, right? I've not listed every single medical condition which is governed by a metal. This is just a handful of a few. let me see if it goes. Yeah. The second category of metals are known as the exogenous metals. So unlike the endogenous metals, these are the metals that shouldn't really be inside our body at all, right? But we're exposed to them through various routes, and that can be several different routes as indicated here. So this could be routes from environmental pollution. So the air that we breathe or the food that we eat, for example, I'm in London, I go on tube, so I breathe in all the horrible air there. Or if you eat things like apex predators with lots of mercury in, things like shark or fish, or like things like a tuna fish, then you are likely to consume quite a bit of things like mercury. We don't want these in our body because they're very bad for you. So there's a big push on the scientific community to try and understand the implications of environmental pollutions in human health. On the absolute flip side, you have the therapeutic metals. So these are metals that we introduce into our body on purpose. So things like therapeutic drugs, like metallo drugs, cisplatin, lots of chemo-based therapeutics are metal-based. But also things like silver is used a lot in antimicrobial resistance, and lithium is used a lot in bipolar disorders. And there's lots of other different metal drugs as well. So these are metals you introduce on purpose. Then we have the group of metals known as the diagnostic metals. So these are things to aid contrast in lots of imaging techniques used in clinical settings. So things like gadolinium and bariums used in things like MRI contrasting agents. We have PET nucleotides like technetium and things like that. And they're used to just help for diagnostic reasons. And finally, here we have the biomaterials. So this is where I originally started. Joanna alluded to this in the introduction. So this is where I originally started my research career, understanding the implications of how metals got released from biomaterials and how they are important in the body. But ultimately, you put a biomaterial in the body to help the functionality of the body. So, for example, if your hip is broken, you might have to replace it with a hip replacement, and then therefore you can walk again, which is great. But unfortunately, these can lead to deteriorating metal particles that release into the surrounding tissues and cause problems later on. So they're all known as exogenous metals. So just to really quickly recap on metallomics, you have the endogenous metals. These are inside us, certain concentrations, and dysregulation causes disease. And then you have all the exogenous metals that are not meant to be inside us, but we're exposed to them through various routes. And all of these metals is the field of metallomics, and how they are influenced. How this influences human health is the field of metallomics. So that kind of summarizes metallomics in a nutshell. And now I want to talk a little bit about the instruments that you need to answer questions on metallomics and some of the stuff that we have at the LMF before moving on to a few case studies. So when people think of metallomics, this is probably the gold standard technique that most people think of. This is called solution-based ICP-MS. So ICP-MS stands for inductively coupled plasma mass spectrometry, which is a bit of a mouthful. But it's very standard. We can look at the bulk level of metals or elements in biological samples, and biological samples could be things like tissues, so tissue biopsies. It could be cells, so a big cell population, fluids like blood, urine, those sorts of things, and we can do this at extremely high sensitivities. So this technique is one of the highest elemental sensitivity techniques. In some cases, we can actually go down to parts per quadrillion, which is really unfathomable, to be honest. Has a very high throughput, so you can get through hundreds of samples in a day. It's extremely robust, and it's a well-established technique. It's pretty much every single university has ICP-MS. They're not that unique in that sense. It's used sort of ubiquitously not only from a scientific and a foundational science perspective, which is what we do here at the MF, but it's also used in diagnostic applications in a clinical setting as well. So I've mentioned earlier about hip replacements. So for example, if you wanted a proxy of how well a hip replacement is doing inside a patient, you could take some blood from their arm and then have a look at how much cobalt they have in their blood. And that is a proxy of how much the implant is deteriorating over time. So there are certain clinical applications for solution-based ICP-MS. So a sample is taken from one of those I've just mentioned. It's digested, usually an acid-based digestion is preferred, into a very, very clean solution. And then from there, it's introduced into the ICP-MS itself through some clever introductory mechanisms. And I'm not going to go into the purpose of this talk today is not to tell you how an ICP-MS works, but just to say you introduce a sample and it tells you what's in it in terms of that periodic table, any elements that you're interested in, extremely high sensitivities and high throughput. The next technique I want to talk to you today about is single cell ICP-MS. So whilst I said previously on the previous slide that we can take a bunch of cells, usually, hundreds of thousands of cells, digest them and work out how much metal's in a specific cell population. Now we'll go in and go, how can we do this at a single cell level? So looking at the metallome of a single cell, right? So in this way, I'm not going to go into super details of the technical aspects again, but effectively what we see here on this slide, and hopefully this plays, great it is for the first time. We see a slide and we see all these little black dots on here are T cells. And what we're doing is we're using a laser ablation system to introduce one cell at a time. So each cell is being introduced, and if I draw your attention to this graph on the top right, you're seeing, in this case, this person was interested in iron. So you see background levels of iron fluctuating, which is just normal background from the instrument perspective, and then you see a single cell event. So this peak here refers to how much iron is within a single cell. If you then fire lots and lots of times at loads of different cells, then you can get really high statistical numbers between cell populations. But specifically, you're looking at single cell level of the metallome, which is extremely cool. And this is actually very, very new to our facility. I think it's probably, we did our first successful experiment around three months ago, which is very cool. And then I jump ahead onto this one. So the next technique and the last one I want to talk to you today, which is probably what we're most renowned for globally, is laser ablation ICP-MS. And talking to a Euro-BioImaging audience right here, this is the one I'm going to focus on for the rest of the talk because it's all about imaging, as the name suggests. So just to explain a little bit about how laser ablation works, because then you'll get an idea of what the data looks like. You have a tissue which is very similar to normal histological prep. So formalin fixed paraffin embedded samples or frozen sections, which are cut to a very thin section. So very similar to normal histology. They're placed on a glass slide. And then you take a very focused laser beam, which then rasters across the sample itself. In this kind of very similar to a sort of a confocal microscope setup, but instead of shining lights or different wavelengths of light at the sample, you're firing a destructive laser path, which then interacts with the sample below and causes an ablation event. And what an ablation event is you can imagine, I know it's quite small, but you have a picture here on the top right where particles are being shot off the sample. So a laser comes down, hits your sample, and material gets ablated from it. It puffs up, and then the material is then in the air above it. We then collect this with some really clever helium tornado, which is really cool, but I'm not going to show you that today. And then this kind of material from the sample is then really quickly transferred to a mass spec. So we have an event where a laser is fired, material comes off the surface of the sample, and then that material is then transferred into a mass spec, and then the mass spec tells you what's in it. Because we can do this with really high sensitivity mass specs and time of flight mass specs, we have the flexibility to image like single isotopes all the way to the whole periodic table at the same time. And what this does is it allows you to produce beautiful images like this one you can see at the bottom right here. So if you want to see what laser ablation looks like on a tissue sample, so this is just a tissue sample, I think it's a breast tissue, and you can see the square, the little red square in the center of the image is the size of the laser spot. So in this case, it's around 20 microns in size, and it's rastering across that tissue itself. What it's doing is it's kind of removing the material beautifully. It's hoovering the material up, and then that material is getting transferred into a mass spec, and then it tells us what's in it. Because we have the timestamp data of when the laser was fired, and also the timestamp data of when the mass spec gets the information, with some clever maths, we can then produce an image by reconstructing those two bits of data sets. And then we can produce images like this. So it's just here, we're just scanning through different elements from a mouse embryo. So things like we can see here, the heart here pops out in an iron signal because you can expect there's lots of blood in there. Things like the lungs and the liver, you can see the small individual vertebrae within this mouse. You can see the brain there and the mouth and lots of different anatomical features lighting up for different metals, which we would naturally expect. So that's laser ablation. This is quite a unique technique. I'd say only probably a handful of groups do this globally, and we are one of them, which we specialize really specifically for biological samples. So this technique has actually been around for quite some time. It was well established in the geological space for people looking at rocks and things like that. because generally people wanted elemental information about rocks, but they're very hard to digest. So then they use a laser introductory system to introduce the sample into the mass spec. And now it's been really exploding in the biological sphere because now we can start to image extremely quickly by and generating metallomic data imaging sets. Cool. And if I jump ahead onto the next slide. I wanted to include this slide because of the audience here, just to give you some facts and figures about sort of what you can expect. I like to call it the top trumps of laser ablation. It's like it's facts and figures. So in terms of a spatial resolution, we're down to the cellular level. So we're around 1 micron in size up to tissue level. So we have that flexibility to change the resolution of the instrument. This is done by actually physically changing the size of the laser. So we can image at a single cell level. Okay, we're not like an electron microscope, but we can say maybe a few pixels per cell down to, but also doing the really high field of views by imaging a whole sample on a whole glass slide. So that's very cool. The sensitivity is in the parts per billion range, so it's not quite as high as the solution-based mass spec, but it's still extremely good for an elemental imaging technique. It's probably one of the best. Speed is very quick, around 5 milliseconds a pixel. So an image like you saw previously, that embryo of the mouse was probably only took around an hour to 2 hours to measure. Its detection, all of these blue elements here, you can see they're all detectable from a mass spec perspective. So you can almost detect the entire periodic table, with some of the exceptions for the really electronegative elements out there, which are very hard to ionize. And then in terms of its output, we are producing elemental images which are quantitative. So not only are we saying, the spatial distribution in counts from a detector perspective, but using some very clever calibration systems, we can then convert that into femtograms, or for biologists, ppm, which is what most people want. So this is to give you an idea of sort of a whole tissue image. At 30 microns, this might have taken sort of 30 minutes to measure. And then you can really focus on specific areas on serial sections, I should say, because this is a very destructive technique. Once you've imaged one sample, it is gone. So then you use the use of serial sections, and then we can use really high resolution things to get fine detail. And this next slide, if I just pop it on, is just to show you some really cool applications that we've been done over the last sort of year or so. So just to explain a few of them. So here we have muscle fibers. So this particular person's interested in aging and physiology between exercising and non-exercising participants, and whether their metallomic profile changes. It's very cool. Zinc is an interesting metal in this case because it's upregulated in fast twitch muscle fibers. So you can almost predict whether this person's an exerciser from their metallomic composition, which is, I think, really awesome. We're doing a huge project with the TNBC from Maddie Parsons, which I'll talk to you about a little bit later on. Inflammatory diseases like rheumatoid arthritis. I've included tissue microarrays here just to show that we can do really high throughput stuff. So we can image sort of hundreds of patients a day within these types of tissue microarrays. Of course, cancer is very important. TB, I'm going to cover a little bit later. But yeah, things like diabetes as well. Zinc is incredibly important in the production of insulin, neurodegeneration, stroke, and things like that. So what I'm trying to show you with this slide is not only just to impress you because the images look cool, but also that there's a real wide range of biological necessity to look at these elements in human health. Cool. So now I've explained kind of what metallomics is and the techniques. The last part of the talk, I want to now talk about some case studies of where we've done it more specifically. So the first one I want to talk about is in infection, and we've been working on tuberculosis for around a year now with a really good collaborator of mine from the African Health Research Institute, Professor Adri Stein. And TB, of course, is still a massive problem in sort of third world countries that lack the healthcare to fight the bacteria. In the UK, I'm obviously very grateful to my healthcare provider. If I got TB, then I would just go and get a strong course of antibiotics and everything would be okay. But for third world countries, this is a massive cause of death. So it's still super relevant to be doing research on this field. A lot more people that you might know actually have TB present in our body. I think it's 1 in 3. Don't quote me on that, but quite a few people actually have TB, but it's sat in your body completely latently. Your immune system is suppressing it, and it's not causing any sort of problems that you would have to go to the hospital about. But it's when TB actually becomes active, it starts to replicate and it causes massive problems. And without realistically good medical intervention, you are going to die from it. So it's still very important to look into. And of course, I'm interested in metals. Bacteria also need metals to survive. They're massive scavengers of things like iron. They need iron to replicate and survive. So obviously, there is an aspect of infection when it comes to metallomic research as well. So you can see some of the images of some patients. These are all post-mortem patients of lungs from people in South Africa from the collaborator that I mentioned. And you can see they have these structures called granulomas. And I'm just identifying one here, which is an image you saw earlier. And you see these granulomas are structures where the TB is present. So if I show you on the next slide, this is just one typical one. So we're employing this sort of time of flight based approach to look at the metallomic imaging of these granulomas themselves. So by using time of flight, we can capture the entire periodic table, not just iron. And there's really interestingly, there's a wide range of metallomic profiles in these types of granuloma structures that I've talked about here. The granuloma is an interesting one because you have in the center of it, you expect to see the bacilli, so that bacteria, and then there's a really weird and wonderful immune system, immune cell population kind of encapsulating it to prevent it from spreading and trying to kill the bacteria itself. The last line of the patients is to completely calcify these and they become very necrotic as well. So what's super interesting in this is we see these metallomic signatures of these granulomas and they're not the same. What was very cool is that if you look at all of the patients that we've looked at and actually several granulomas in a single sample, there's different metallomic signatures in each one. Now, what we're speculating is actually the body is trying to do some really clever things by using metals to their advantage to try and kill the bacteria. So what we see in a lot of them is actually inside the granuloma, where we expect to see the TB bacilli, is they are starving them from iron. The bacteria need iron to survive and replicate. So why don't we just completely remove the iron from that microenvironment and hopefully it should kill them. On the flip side, we see massive concentrations of things like copper and manganese and zinc, which are at concentrations which could theoretically be toxic. So it could be that these immune cells are not only sort of sequestering this metal from the environment from iron, but they're also dumping other metals to try and make a toxic environment. And this data is really recent, and the story keeps unveiling as we're looking at it. One thing also to plug here is that we are doing time of flight technology. So we're not just interested in one or two metals, but we're interested in kind of the whole periodic table, not including also including the non-metals as well. So this graph here, you can see it's quite small, but along the x-axis here, you've got all the metals, and along the y-axis, you've also got the metals. And this is just a linear correlation between them. So you can see, for example, things that are red says, so for example, this is strontium, and it's highly correlated with lots of different metals. Now, this is very cool because now you can start to unpick not just single metals. So, for example, as iron upregulated or downregulated in areas, but now you can start to pick whether there's relationships between metals. And actually, that starts to open real new mechanistic understanding between why metals are correlating or co-locating with each other, which is very cool. I'd be super interested if anyone has really cool ideas or ways of doing things with not just two metals at a time, but lots of different metals and understanding signatures. Please hit me up because I'm willing to learn new techniques as well from the analysis side. So that's from the TB case. So from the flip side, this is a little bit more about my background. So on exogenous implant debris. So this is from the exogenous metal experiments. So this is from a bone anchored hearing aid. So this is a very small titanium screw. It's only sort of about 3 or 4 millimeters in size. It's placed post-auricularly, so it's just placed behind the back of the ear. A microphone is then placed on it, and then it allows some signals to be placed into the ear to allow hearing to be reestablished to people with conductive hearing loss. The issue with these screws is, well, normally they're very good, but one problem of them is they can release these metals from normal routine clinical service, and then these metal ions can then accumulate in the surrounding soft tissue, so the kind of the skin that's around the back of your ear, and cause, unfortunately, lots of inflammation and a lot of pain, and ultimately leading to revisional surgery. So I've been working quite a number of years with Professor Owen Addison at KCL on these types of implants and trying to understand what is released from these implants and what sort of immunological responses is happening from them. So these particles, unfortunately, you can't see them in routine histology because they're too small, or they don't have they're lacking contrast. So we have to use this elemental imaging techniques to actually look at this titanium itself. So these implants are made from commercially pure titanium, so should only have titanium in themselves. So we can see the distribution of this, it's incredibly heterogeneous. So it's not just a homogeneous blur of titanium, but there's lots of little particulates that have come off. Some of them are very large and some of them are very small. With some sort of complementary technology, you can also understand the chemistry. So you can use things like X-ray spectroscopy at synchrotron-based light sources to then understand, okay, what's come off? Is it titanium? Is it metallic? Is it an oxide? Is it a phosphate? And why is that important? Is because the chemistry of metals is equally as important as the presence of them in the first place, right? Think about it like this. Chromium 3, an oxidative state of chromium 3 is completely normal. Chromium phosphate, it's very, very inert, doesn't really do much to your body. But hexavalent, so chromium 6+ ions, are extremely carcinogenic and will give you cancer. So chromium itself it's both present as chromium, but the valency or the chemistry of it is very important. So that's a reason why when we're looking at exogenous metals, we should also be thinking about the chemistry of them as well. This panel here is just to show that we've done things at different resolutions. So from sort of single digit micron numbers all the way down to sort of 50 nanometer resolution. And this panel really is just showing that we see big lumps of metals into tens and 20, 30 micron lumps, but also we're seeing single things which we know we can resolve to around 50 nanometers. So we're seeing this real wide range of particulates that are released from these implants. With some sort of serial histology, we can then sort of link this to the immune system, where these correlate to certain immune cell markers like macrophages, T cells, and plasma cells, and showing actually we see a positive correlation between titanium that's released. We see the little particles causing a massive immune response. And yeah, so then we can start linking the mechanisms themselves. So I like to throw a little mechanism in there just to say we don't just look at pictures, but we're trying to understand the science behind it as well. So this is just the release mechanisms. How is the metal come from the implant itself? So some sort of types of biocorrosion, cellular biocorrosion, biofilms in part of it, and sort of vibration of this instrument's causing some sort of articulating forces, which is causing the release of particles themselves. Those particles can come off as ions, which are extremely reactive, and then they precipitate immediately, or they can come off as physical particles from the implant themselves. Ultimately, depending on how they come off it, they dictate whether they get associated with macromolecules or proteins, or whether they're taken to the bloodstream and then they're systemically or excreted through your sort of normal routes as well. So why is all of this important? So one, it sort of guides our perceived biocompatibility of implants, whether we should be putting these in patients or not. Hopefully, it can contribute to sort of the design of the implant. For example, I've had cases where you have a lot more metal release from implants, and that's maybe because the implant surface has been changed, and maybe that's causing release of more particles, which might be eliciting a further immunological response. And also, if this guide to tell us when we need to make a surgical intervention for that patient as well. So that's kind of some of the reasons why we want to do this sort of work as well. So finally, probably one of the biggest studies that I've done so far is with Professor Maddie Parsons here at KCL on the triple negative breast cancer story, which was funded by Wellcome Leap. So for those who don't know, TNBC accounts for around 15% of all breast cancers. It's more common in younger women than older ones. It's unfortunately highly aggressive and has a poor prognosis, often leading to death within the first 2 years, which is of course not a good thing. for me to explain kind of the rationale of this project, it's easier for me to go through like a sampling strategy. So a patient comes in, they have been diagnosed with TNBC. They have a thing called a needle biopsy. So a needle is placed inside the breast, it hits the tumor. So a very small slither of tissue is taken out. It almost looks like a little worm of tissue, very, very small, around 1 millimeter in diameter and a few centimeters long. That patient then has just normal routine neoadjuvant chemotherapy. And then what we have is we have the understanding of whether that patient responds to chemo or they don't respond to chemo. So we have a sample before the patient has their normal therapy, and we have the understanding of whether they respond or not. Unfortunately, so if they respond, that's all good news. Unfortunately, if they don't respond, then they have to have some sort of surgical intervention where a large piece of the tumor is removed, if not an entire mastectomy at that point. So we have this really cool bit of data set because we have a sample before they have treatment, and then we have we know whether they responded or not responded. Now, why are metals actually important in cancer? I could spend a whole hour talking about this, but very briefly, iron is incredibly important in things like oxidative stress, the production of these pesky molecules called reaction oxygen species, which go around and cause lots of DNA damage and mutation. Things like zinc and magnesium, super important in cell signaling, division, telling cells whether to divide and replicate, and growth through various cell pathways. Magnesium, incredibly important in immunity, and copper, important in things like the process of angiogenesis, which is the formation of the blood vessels. So you can imagine cancer needs quite an intricate blood supply to provide nutrients and oxygen to the growing tumors. So the idea, or the whole crux of this kind of story that Maddie's been doing, is can we look at markers at this point to understand whether we can predict whether they're going to respond or not? Because if we know they're not going to respond, then don't put them through chemo and go for a different surgical route or different treatment option. So we have the sample. We're looking at these specific metals because of their importance in cancer. And like typical Maddie fashion, we have imaged a lot of samples. So we've imaged probably the biggest elemental cohort of patients to date I know of globally, which is quite difficult because it's a lot of samples. These things take a lot of time, and we learned a lot from the first sample and the last sample we ran, because you can imagine this is collecting data over several years, which is very challenging. And from a mass spec side, making sure the way you've run the first sample and the way you've run the last sample is the same is extremely difficult, because you can imagine things have changed within 2 or 3 years, not in terms of just the technology, but the analysis pipelines and everything like that. So it's been a baptism of fire, and I love this project and hate it at the same time. So a little bit about the results from this. Still really ongoing. As you can imagine, it's a very, very large data set. But from the metallomic data set alone, we are seeing differences in those key metals that I mentioned, and the predictive power of effectively predicting whether a patient is going to respond or not respond just to metallomics alone has been really successful. I should also say this has been made entirely possible with an amazing collaborative group from the BGU University in Israel, who have taken on a lot of the data analysis and these predictive models to understand whether metals alone and lots of different modalities can predict whether patients will respond or not. So takeaway home today, message on this bit is that there are definitely different metallomic signatures. We're still trying to unpick the mechanisms why there are differences. And maybe there will be different diagnostic implications just on metals alone. Again, in Maddie fashion, she threw the whole arsenal of different modalities at these samples. They're incredibly precious, and they're a really cool cohort of samples, understanding whether they've responded or not. So there's loads of different techniques that have been thrown at these. And other than just the laser ablation, which is obviously what I've shown you just a second ago, but there's a real nice correlation with imaging mass cytometry, which I just want to go over briefly now. So just to recap on imaging mass cytometry, this is an imaging approach where we can label using antibodies, but instead of bound to traditional fluorescent probes used in sort of confocal microscopy, we bind lanthanide tags or rare earth metals. What that allows us to do simply is flood a whole tissue with 35, 40 different markers at the same time, and look at all of these different protein markers. Some of them are immune cell markers, tumors, and stroma material. All of these markers can be imaged simultaneously, which is an extremely cool technique. So from this sample, a sample is taken, a slice is taken for laser ablation. So we're seeing those really critical metals that I've talked about all day. And then also a serial section is taken for imaging mass cytometry to allow you to image all of those different protein and cell markers on a serial section. Now, when we start to do correlation between these, this is the really exciting part, because now we're starting to pick correlations between metals and protein markers on the same on these data sets. So I know this is a bit of a confusing graph, but if you just draw your attention along the Y, we have different protein markers from that imaging mass cytometry. And along the X, we have our different metals that I've talked about today. And things that are red are positively correlated. So you'd expect to see as one protein goes up, the metal goes up, and things that are blue are negatively correlated. So the inverse of what I've just said. Now, if I just draw your attention to one correlation, just to, because I can't go through them all today. But for example, here we have a CD68, which is a macrophage marker. We're seeing it's extremely positively correlated with iron because it has a very high linear regression score, which makes biological sense, right? Because we know macrophages love to sequester iron inside the cytoplasm of the cell as ferritin. So you would largely expect to see where you see macrophages, you also see iron. Now, the cool part of this, of understanding a relationship which you can explain biologically is fantastic, because it shows you that the technique is working. But more importantly, what this allows us to do is between the responders and non-responders, see not only are there differences in their metallomic profiles, but also whether there's differences in the relationships between metals and cells. For example, if you have too much iron or too little iron in a macrophage, they don't work. So that might then be an indicator for why we're seeing response in not just in cancer, but in any plethora of diseases you can ever imagine. I like to throw this kind of graph here is that metallomics of what I've shown you today is really looking at the constituent elements of everything. It's the fundamental building blocks of life, and it's the very starting point, the atoms and the elements. But on the way to the human body, there are lots of different things, metabolites, proteins, cells, etc. And all of these are equally as important to study. So it's not good enough nowadays as data scientists or biologists to look at these as in silo alone, and that's why we need these correlative analysis to be able to pinpoint, for example, not only just the metals, but the relationships between cells and proteins as well. So I just wanted to leave this kind of message to you that I think this, from my experience over the last sort of 3 years in this role at the London Metallomics facility, these relationships are starting to unpick sort of more mechanistic understandings of metals rather than just looking at a pretty picture of metals on their own. I'm just going to look at my time to see. Yeah, so what I'm going to do, I'll leave it there because I want to give the opportunity for people to ask any questions. I'd like to thank everyone for listening and showing up today. Thank my team at the LMF massively for supporting all of this sort of work. And the team, Adri Stein at AHRI and the BGU team in Israel are fantastic collaborators to work with on this sort of adventure. If you are interested in doing any metallomics-based research, we are super open and collaborative to do some work together. So please contact me via this email or scan the QR code. And thank you for having me.